Go ahead and get started. Okay, good morning, everyone. Thank you all for coming. And I want to thank and introduce our speaker for today, Dr. Stephanie Bissel. And Stephanie is currently an associate research professor in the Department of Medical and Molecular Genetics at Indiana University, and also works with their Alzheimer's Disease Research Center there. Stephanie got her PhD in neurovirology at University of Pittsburgh and then completed some postdoctoral training at Pittsburgh, as well as at UCLA. So uh, Stephanie is currently one of the principal investigators on a grant looking at the impact of PLCG2, an Alzheimer's disease risk variant, on microglial biology and disease pathogenesis. Also works on the project, large project, multi-center project, looking at connectopathies at cellular resolution in the aging and AD mouse brains. So we're very happy to have Stephanie here to talk about her work on PLCG2 and Alzheimer's disease. So thank you.
Thanks for that nice interjection. I feel free to speak up and interrupt me at any time. I have no problem with that. So here are my disclosures, but this has no impact on the data I'll show you today. So I'm sure you're all familiar with this particular graph here, but what I kept it in to show you the green line here. So this is the microglia responses that largely parallel amyloid pathology. So we know that microglia at least have some role in the progression, maybe the development of Alzheimer's disease. And if we look at microglia responses pathologically, we see the A beta plaques in red. The green is the IBA1 positive microglia. And you see that those microglia surround the plaques and engage them. So looking at the genetic landscape over the last 15 years, the GWAS studies that have been, the large GWAS studies that have been published have really highlighted the role of microglia in the pathogenesis of Alzheimer's disease. Several of these, about 40% of these GWAS hits, are exclusively expressed in microglia in the brain. And if we look at where a number of these GWAS hits, if we look at microglia versus total brain lysates, we see that many of these are enriched in microglia. And they have mostly, to do with innate immune responses, but also in the endolysosomal network and phagocytotic responses, as well as the lipid and cholesterol metabolism. So what do microglia do? Primarily, these two functions, phagocytosis and inflammation, are the ones we think of most, but they have a number of other functions, from metabolic responses to oxidative stress. They support remyelination and have a large role, especially in development in synapse surveillance and pruning. But this also probably comes into play later in neurodegenerative diseases. One thing that we'll talk about a little bit more later is that once the microglia encounter an insult, whether it be viruses or protein aggregates like amyloid beta or tau, they adopt a disease-specific signature. So this is mainly right now defined on transcriptomic signatures, but there are different types of microglia depending on these insults and the region. So many of you probably have heard of the disease-associated microglia. This is one type of that genetic signature that is a response to Alzheimer's disease pathology, and we'll touch on this a little bit more later, but I just wanted to draw your attention again that microglia generally engage with the plaque, form a barrier, and once these microglia that do this are thought to be protective. They change their transcriptomic profile. They enable the microglia to cluster and compact the plaques more and maintain metabolic fitness. So back to this genetic landscape, one that I want to point out to you that caught our eye a number of years ago is PLCG2. So PLCG2 was identified in 2017 with this variant P522R, and it was a protective variant. So we were somewhat interested in that since we had been studying TREM2 for a while, which was a risk variant. It was an interesting sort of protein. It the carriers of P522R exhibit a slower cognitive rate of decline. And interestingly, it's also associated with protection from FTD and Parkinson's disease, and also is associated with longevity. Functionally, it's been shown that this variant has a slight hypermorphic activity. Whether that plays a role in its protection is still to be debated. So with all this information, we thought that PLCG2 may play a role in AD pathophysiology. And so in collaboration with the Model AD Center, which creates new models of mirroring Alzheimer's disease, we did some analyses and found that another variant of PLCG2, M28L. And this one, which is shown here found in the pH domain, confers a slight risk for Alzheimer's disease. And we see that the protective variant is found between the split pH domain and the NSH2 domain. And these ribbon cartoons show the approximate location. They're found in different domains of the protein. And as I mentioned before, the protective variant is thought to have a slight gain of function, whereas we have shown, and I will show you, that the M28L variant seems to have a loss of function. So what is PLCG2? So primarily PLCG2 is an intracellular signaling hub. So it's downstream of a number of innate immune receptors, including the Fc gamma receptors, CLEC7A, and the toll-like receptors. But what I'm showing you here is it's downstream of TREM2, which is what I like to call the poster protein for microglia risk factors, and is heavily studied in the microglia responses to Alzheimer's disease pathology. It recognizes lipid moieties, and once it is engaged by these moieties, it partners with its co-receptor DAP12, since TREM2 has no intracellular signaling domain. And this leads to a cascade that recruits Syk, phosphorylate Syk, which can go on to phosphorylate PLCG2, as well as other adapter proteins that are associated with PLCG2. And this enables PLCG2 to move to the membrane, where it engages its target PIP2, and it hydrolyzes PIP2 into DAG and IP3, which then activates calcium responses and transcription of immune response genes. So today I'm going to show you some small and some larger stories about our research in PLCG2 and AD. So first we're going to look at some human expression of PLCG2. We're going to look at the 5X FAD amyloid mouse model that has been crossed to these PLCG2 variants. We're going to look at what happens in the absence of PLCG2 and whether this is similar to what happens in the absence of TREM2. And then we have just begun using some iPS-derived microglia to dissect the molecular mechanisms behind the differences in these variants. So first I'll show you the human data, and this was led by a graduate student who's now a postdoc at Stanford. And he took the bulk RNA-seq data from the AD knowledge portal and queried a bunch of different brain regions to see what the expression is like in each of these brain regions. And many of the cortical regions and hippocampal regions showed that with Alzheimer's disease, we had an increase in PLCG2 expression. And this increase correlated with the mean plaque density. So we wanted to see whether this held true, since we are a mirroring lab, held true in the 5X FAD model, amyloid model, and we see a disease progression increase in the 5X FAD PLCG2 RNA levels. And we also wanted to attribute this to make sure that their microglia were the cells that were responsible for this increase in PLCG2 expression. So we, at 4 months of age, we treated these mice with Plexicon, which is a CSF1 inhibitor, which depletes microglia. And after 28 days of this treatment, we looked at PLCG2 expression levels, and we see that in the absence of microglia, that the PLCG2 expression level is down to baseline, which suggests that this increase we see is due to the presence of microglia. Back to the human data, we also wanted to see what happens with cognitively normal aged individuals with low or high PLCG2 expression. So we ranked PLCG2 expression in the AD knowledge portal databases from low to high, and we took the bottom 25% as low expressors and the top 25% as high expressors. And when we looked at the difference in the low versus high, this volcano plot shows you that there are several down-regulated genes in these low expressors, and these primarily had to do with the inflammatory response. So onto the mirroring model, where we crossed the 5X FAD mice with the variants that were expressing, or the mice that were expressing the PLCG2 variants, and we looked at 2 months of age, at 4 months, and at 7.5 months. We start to see a decent amount of amyloid pathology in these mice at this time, and it's pretty substantial at 7.5 months, and I'm going to focus primarily on the 7.5 months. And again, this work was also done by Andy again. First, I like to show this because I'm really, it still astounds me. We collaborated with Nian Wang to image these mice in what was then our brand new 9.4 Tesla small animal MRI. And he had a feeling that we could image plaques, so that he hypothesized that the hypointense areas would correspond with amyloid plaques. And we did some follow-ups to these images and with immunohistochemistry, and indeed it did seem that these hypointense areas correlated with the plaque locations. And so what we can see is that the protective variant has less of these hypointense areas than the wild type 5X FAD model, whereas the risk variant seems to have more. So we looked at this using standard methods of immunohistochemistry, where we stained brain slices for NUN, 6010, and X34. And this shown here is in the subiculum. And we measured the area of the plaques. So the 6010 plaques, positive plaques, are thought to represent the diffuse plaques. And we see that the M28L risk variant has increased plaques, whereas the protective variant has decreased plaques. Similarly, for the M28L risk variant in the X34 compact plaques has increased plaques, but we seem to see similar amount of compact plaques in the protective variant. So we wanted to see if this change in plaque pathology affected behavior and synaptic function. So first we looked at working memory by performing a Y-maze alternation assay, and we see that compared to normal black 6 mice, 5X FAD mice have a decrease in percent alternation. The risk variant seems to have maintained this decrease in percent alternation, but the protective variant rescues this decrease in working memory. So because of this, we wanted to look at if there were differences in the synaptic function. So we looked at long-term potentiation, and we see again that 5X FAD mice have a decrease LTP compared to the B6 mice, and that if we look at the protective variant, that it rescues LTP. And this is just quantifying here the last 10 minutes, and similarly we see that rescue in the protective variant. So what's happening with the microglia? So we stained these brains with IBA1 to look at microglia, X34 to look at plaques in blue, and then 2 microglia markers, which are difficult to discern in these overlaid images. But I wanted just to concentrate here on the IBA1 positive green cells. And we see that in 5X FAD, that there is a response to the interaction of microglia with the plaques. But if we look at the risk variant, Andy always liked to say that the microglia were lazy. They didn't seem to, they ignored the plaque, they seem to ignore the plaques much so more than the 5X FAD. And then if we look at the protective variant, there's potentially an increased engagement with the X34 positive plaques. So we quantified this, and first we looked at IBA1 showing all the percent area in the subiculum. There seemed to be no difference in overall IBA1, but if we looked within the area of X34 plaques, we see a decrease in M28L and a slight increase, although not significant, in the P522R animals. If we look at CLEC7A, which is the marker of the disease-associated microglia and an activation marker, we see overall that both variants have a decrease in the overall subiculum. But if we look within the area of the plaques, there's a decrease in the risk variant, and similar to a slight increase in the protective variant. Then if we look at P2RY12, which is a marker of homeostatic microglia, we see that the risk variant increases overall homeostatic markers in the subiculum compared to the other two, but within the area of the plaque, there seems to be no difference. So we looked at overall cytokine concentration. So one caveat here is that this is total brain lysates. It's not specific to microglia, but microglia are thought to produce the majority of these cytokines. And we saw some interesting results that we're still trying to wrap our heads around because it was much different than what we expected. And I'll take IL-1 beta as that example. So IL-1 beta is generally thought to be chronically a bad thing for the brain, so that many, hundreds of papers cite it as being a neurodegenerative cytokine when expressed chronically in the brain. But when we look at our levels, we see that the protective variant has increased IL-1 beta expression or secretion in the brain, whereas the M28L has slightly less. So this was difficult for us to comprehend, but we have actually recapitulated this in a couple of other models that I won't show you today. So we think this observation is probably real, but we're planning to hopefully work this out in the iPS-derived microglia. We see a similar trend for TNF alpha as well as CXCL1. When we look at we use a MARS to look at potential internalization of A beta. The IBA1 microglia marker is represented in red, 6E10 plaques in gray, and the blue shows co-localization. So this is in the literature usually indicates that this means internalization, but whether it's internalization or just engagement with the plaques, I think is debatable. But we do see that the risk variant has decreased co-localization of IBA1 and the 6E10, whereas the protective variant has increased co-localization. So to see if this had anything to do with phagocytic responses, we created primary microglia from these variant mice, and we fed them with Frodo-labeled A beta, and we see that the protective variant has less fluorescing A beta in the lysosomes of these microglia compared to both the 5X FAT and the protective variant. And this is quantified here, showing a decrease in the protective variant or in the tetramental variant and an increase in the protective variant. So we wanted to see what could be accounting for all of these changes we saw in microglia. So we undertook single nuclei RNA-seq and with a focus on the disease-associated microglia. So I mentioned before that these are microglia are the ones that are responding to the plaque. So generally microglia at the transcriptional homeostatic level express P2RY12, CX3CR1, these types of microglia are thought to be maintaining not only homeostasis, but neuronal health as well, and surveying the brain. But upon insults, whether it be apoptotic cells or A betas species that interact with TREM2, you can get these intermediate states of activation that can become the disease-associated microglia that are dependent on TREM2 signaling. And some reports also indicate ApoE signaling. They are positive for these transcriptomic markers and are associated with metabolic fitness and microglial clustering. There are also other flavors of microglial transcriptional signatures. So one is shown here is the interferon responsive microglia these types of microglia are thought to be the detrimental microglia that are associated with synapse loss and the increased pro-inflammatory cytokines. And being the good immunologist that we aspire to be, the field has made a plethora of these types of cells. So there's DAMs, WAMs, PAMs, and several others, but they are all characterized by increases in some transcriptional markers and decreases in others. So to get at this, whether the PLCG2 variants are changing these, the frequency of these protective microglia, we did single nuclei RNA-seq, and this UMAP shows you the populations of the different brain cells that we identified and the frequencies of these nuclei within each group. We do see some differences in the percentage of microglia nuclei that we see between the groups, but we are unsure at this point what this might mean. So if we focus on the microglia population and perform UMAPs analysis on that population, we identify 5 different subpopulations of microglia. And if we just look here at the risk variant, this 0 population is associated with the homeostatic signature. It seems in the risk variant that this is similar to 5xFAD, but in the protective variant, this is decreased. If we look at the transitional microglia signature, the risk variant is able to increase this signature similarly to the P522, the R protective variant. But it seems to get stuck here because then the disease-associated microglia signatures indicated here as activated A and activated B are decreased in the risk variant, but are increased in the protective variant. So this also supports the idea that these disease-associated microglia are protective. Interestingly, those interferon responsive microglia are increased in both variants, which we were hoping to see a decrease here, but we were somewhat surprised by this, but it's the data. So here are the genes that are associated with each of those activated responsive microglia. These are up in the first population, down in the second, similarly with the B population. And we see that the risk variant has decreases in both, and the protective variant has increases. So to summarize this portion of the talk, we showed you that the disease or the protective variant of PLCG2 leads to restoration of cognitive deficits, increased engagement with the plaques by microglia. There's fewer plaques and increased plaque compaction, and we get an increase in the population of the cells that are responding to the plaques. If we look in vitro, we seem to see an increase in the ability of these microglia to uptake A beta. Conversely, the risk variant shows cognitive deficits, poor plaque engagement, more plaques, and a decrease in these plaque responding microglia and the ability to take up A beta. So we wanted to see what it was the impact of total PLCG2 deficiency on AD pathogenesis. And also secondarily, we added a TREM2 deficient model to answer whether PLCG2 has any functions that are independent of TREM2. So to do this, and remember PLCG2 is downstream of TREM2. So that's why we wanted to see if there are any independent functions. So we took 5X FAD mice and crossed them to PLCG2 knockouts and TREM2 knockouts, and this work was led by Evan Messenger. And there's one caveat with the PLCG2 knockout. We have been breeding these for a number of years, and we've only been able to acquire a handful of PLCG2 knockouts. So the data we have, I will present to you, is only with 6 PLCG2 knockouts on the 5X FAD background. So just to give a sense of what the different functions of TREM2 are, so TREM2 is known to sense lipids and be important in the metabolism. Both TREM2 and PLCG2 are important in phagocytosis, but PLCG2 is important in cytokine production and thought to be important in cytoskeleton rearrangement and motility. And together they promote survival of microglia and lipid homeostasis. So the first thing Evan looked at was the expression of both PLCG2 and TREM2 in these brains. So in PLCG2, not surprisingly, we had no detection of PLCG2 in the PLCG2 knockouts. And in the TREM2 knockouts, we seem to have slightly increased expression of PLCG2. So this is shown here in this graph where we get a decrease in the knockout and an increase in the PLCG2 knockout and an increase in the TREM2 knockout. But surprisingly, when we looked at TREM2 expression, we see an increase in TREM2 expression in the 5X FAD mice, and this is no expression in the TREM2 knockout, but we also see extremely limited expression of TREM2 in the PLCG2 knockout. So this is represented here in this graph showing that the PLCG2 knockout has decreased TREM2 expression. If we go back to our RNA transcriptomic data, we see that this is also reflected at the transcript level. So the PLCG2 knockout mice have decreased TREM2 gene expression. So we wanted to look and see if this was apparent by immunohistochemistry. So we stained the cortex of these mice with IBA1, which represents microglia, TREM2, and TREM2. And we see that, again, that the wild type increases expression of TREM2 associated with microglia. The TREM2 knockout has no expression of TREM2, and the PLCG2 knockout has limited expression of TREM2. So this suggests that PLCG2 is necessary for proper TREM2 induction. We wanted to see if this held true with humans. So we took the data from the Mount Sinai Brain Bank and we compared PLCG2 expression to TREM2 expression, and we either looked at expression based on SARAD scores or by Brock stages. And we see that the PLCG2 expression correlates with TREM2 expression, and this is independent of AD pathology. So the hypothesis that PLCG2 is responsible for TREM2 induction holds in human data. So back to the mice, we wanted to look at how both of these deficiencies affected amyloid pathology. And so we stain the brains for X34 plaques, as well as LAMP1, which is a lysosomal associated membrane protein, which is a marker of dystrophic neurites. So when we analyze this, we see that the total plaque percent area, we saw a trend of increases in both knockouts, but these weren't significant. But if we looked at the average size area of the plaques, this was increased compared to wild type. This was more apparent with the LAMP1 marker, and we saw increases in TREM2 as well as PLCG2 knockouts. If we look at how the microglia engage the plaques in both of these animals, we see that both the PLCG2 and TREM2 knockout have a decrease in both the plaque coverage, the engagement of the plaques, as well as the area surrounding the plaques. So we just took this periplaque area and saw decreases compared to the wild type 5X FAD mice. So when we look at bulk RNA-seq of these mice, we see similar deficiencies of TREM2 and PLCG2 in terms of immune pathways. So both of the knockouts have decreased expression of genes associated with the immune response. And this is shown here in this heat map where you have the wild type 5X FAD versus both of the knockouts that have decreased expression of these inflammatory immune pathways. So at least in terms of the immune pathways at the transcriptional level, we seem to see that TREM2 and PLCG2 are very similar to each other. So we wanted to see if this was true at the protein level. So we selected a couple of these, and we looked at CD11C, also known as ITGAX. It's a marker of a disease-associated microglia, and it's important for cell adhesion, chemotaxis, and antigen presentation. So the 5X FAD has an increased expression of CD11C, whereas both the PLCG2 knockout and TREM2 knockout seem to have limited CD11C expression. If we look at another disease-associated microglia, we finally see a slight difference between PLCG2 and TREM2 knockouts. So here we're looking at CD68 expression in red. So CD68 is a marker of lysosomal activity. So you see that this is increase in response to the plaque in the wild type 5X FAD, but you get a severe decrease in the PLCG2 knockout and a decrease in the TREM2 knockout, but not to the same extent as the PLCG2 knockout. So we analyzed this in a number of ways, total percent area in a relation to the IBA1 positive microglia marker, as well as the periplaque area. And we see that the PLCG2 knockout has decreased lysosomal activity compared to the TREM2 knockout. So because of this, we wanted to see if pathways outside of the immune response that we looked at initially were different in PLCG2 and TREM2 knockouts. So we did a PCA plot, and indeed we do see distinct transcriptional differences where each of these groups cluster independently. And if we look there at the differences, there are about 500 shared genes between the knockouts, about 600 unique to, are enriched with the PLCG2 knockout, and about 400 to the TREM2 unique to the TREM2 knockout. And so if we look at the differences between the shared pathways, the PLCG2 knockout versus 5X FAD, we get an enrichment of cell surface signaling, cytokine production, cell migration, versus the TREM2 knockout, which has an enrichment in cell adhesion and development, genes associated with developmental pathways. So since we have limited number of knockout mice, we wanted to see how we could look at this at a molecular level and a functional level. So that this is where we switched to looking at iPSC-derived microglia. So we took donor microglia from Matt Blurton Jones, and he actually had created the protective variant, and we took that variant and we created a knockout line and a line for the detrimental variant. And this work was led by a graduate student in the lab, Logan Beffer, with help from the Jason Meyer lab, and a student, Kaylee Tutro, and a tech, Carly Hooper. So to get iPSC cells, we are derived microglia. We take the iPS cells and we treat them with 2 different types of media for 12 days to get the hemopoietic progenitor cells. We make a huge batch of these, freeze these down, and when we are ready for the experiments, we take the frozen stock and we culture them with these 3 cytokines, which drives microglial differentiation and maturation over 28 days. So we see with our 4 lines that we are able to efficiently create HPCs, and that the microglia differentiation seems to work with all of these lines. If we look at the characteristic microglial markers at the end of that maturation period, we see that the IBA1 marker is expressed in all the different lines, as well as the homeostatic P2RY12 marker. We are currently, the shapes of these cells suggest that there may be some morphologic differences, and we're currently analyzing different batches of cells to see if this is true. This experiment is just to see if they are competent phagocytes. So microglia are known for their phagocytotic ability. So we hit them with photo-labeled E. coli, and we see that after 6 hours of E. coli, that all of the different lines are able to phagocytose the E. coli. So the first thing after we characterize the microglia of whether these lines were able to create viable microglia, we then did an unbiased basal characterization of these using RNA-seq. And when we did this, we saw that we get distinct clusters of each of the lines, with the knockout and the M28L being very different from the wild type, whereas the protective variant seems to have a lot of similarities with the wild type. And keep in mind, this is at a basal level. So far, that's what we have examined. So the next thing we did was to see if you remember back to the PLCG2 knockout, where the PLCG2 expression seems to be necessary for TREM2 expression, we wanted to see if which genes correlated with PLCG2 expression, and we indeed did find that TREM2 was among these genes. So we looked at the expression of TREM2 in the different lines, and we see that at the transcriptomic level, that the PLCG2 knockouts have decreased TREM2 expression. So this seems to hold true in the IPS model, similar to what we saw in mice. So if we look at the staining of PLCG2 in these different lines, we see that at the protein level, both the knockout and the M28L risk variant seem to have decreased PLCG2 expression. If we look at the same thing with TREM2, we see that there is a slight decrease in the protective variant in TREM2 expression, but this was not significant, and it was not as severe as the decrease observed in M28L and risk variant and knockout cells. So if we looked at what it was differentially expressed between these two genes, you can see that the knockout and the M28L account for the largest number of the differentially expressed genes between the groups. Whereas if we look at the protective variant, there are not very many unique differentially expressed genes at the basal level. If we look at this heat map, it reflects this, that the wild type and the P522R look fairly similar, although there are some slight differences, whereas the knockout and the M20 and the risk variant look a lot different from the wild type and protective variant, and they also look different than each other. So these volcano plots show the differentially expressed genes between the wild type and the other lines. And we see again that the P522R has the fewer number of differentially expressed genes, whereas there is quite a number of differentially expressed genes in the knockout and risk variant lines. And then we looked at the functional pathway analysis, and we see that the protective variant has a unique pathway in the adhesion cluster, whereas the knockout has a unique pathway in proliferation. So we were planning to do some functional follow-up. So today I'm going to show you the proliferation results. We also see that there's an immune component to each of the lines, as well as an apoptosis component. So I'll show you some functional follow-ups to these. So first we'll start with proliferation. We saw that there was increased differentially regulated transcription of genes associated with proliferation. So we did this EDU proliferation assay. So at the end of the microglial maturation period, we fed the cells EDU, and 24 hours later, we used this click-it chemistry to visualize the EDU incorporated proliferative cells. So this was the results of this, and it was somewhat surprising to us. So the wild type and the protective variant looked similar to what we would have expected. The wild types generally do not have a high proliferation rate, and we see that the protective variant is similar to the wild type, whereas both the knockout and the risk variant have increases in the percent of proliferative cells. So this was somewhat surprising to us because we have more trouble growing the knockout and the risk variant lines. But the next assays we did might explain some of this apparent discrepancy. So next we looked at apoptosis. So how we did this is at the end of the microglia maturation period, we deprive the microglia of their cytokines, which is stressful to them and leads to cell death. And so first we looked at the live dead assay. At 0 hours, you see most of the cells are living, whereas at 48 hours of cytokine deprivation, we see that there are many more knockout dead cells in the knockout and risk variant line compared to the wild type and protective variant line. And this is just quantification we did over 48 hours. So at 48 hours, you really see this difference between the groups, and this reflects that final time point. So we did the same cytokine starvation assay that I showed you in the previous slide, but this time we looked at cleaved caspase to show activated caspase and as a marker of apoptotic cells. So after 48 hours, we see increases in the number of apoptotic cells in the knockout and the M28AL risk variant, as well as the wild type, whereas the protective variant is protected from cleaved caspase activity at this upon cytokine starvation. So the final thing I'll show you is we wanted to see how the cytokine secretion differed among these different lines. So we hit the mature microglia with LPS and did a mesoscale panel to look at cytokine secretion, sorry. So we again see surprising results that upon LPS treatment, we see increased secretion of several cytokines in the protective variant. So I've asterisked the different ones. So we see IL-1RA, IL-6, TNF-alpha, CXCL1. So most of these mirroring what we saw in the mirroring line in vitro. The IL-1 beta did not recapitulate, but the other trends did. So even in our IPS lines, it seems that these protective variants are slightly hypermorphic, and we're planning to now dissect the mechanisms behind this as follow-up. So to summarize this data, the IPS-derived microglia, the protective variant has similar TREM2 and PLCG2 expression to wild-type microglia, whereas the risk variant and knockout have decreased expression, suggesting that PLCG2 expression is important for TREM2 induction. The proliferation was similar for the protective and a variant increased in the risk and knockout variants. Cell death was decreased, at least in terms of caspase in the protective variant, and increased in the knockout. And we see altered increased pro-inflammatory cytokines and chemokines in the protective variant upon LPS, whereas it was decreased in the detrimental variant. So overall, what I've showed you is we've taken AD genetic hits, so PLCG2, and did omics data to try to find the transcriptomic and eventually we're working on proteomic difference and metabolomic differences in these different mice and lines, and translate these to the functional and molecular differences so that hopefully we can inform therapeutic targets and processes and really dissect what promotes a beneficial microglia state versus a detrimental microglial state. So I'd like to thank the men who did the work. Evan, Logan, and Andy did the majority of this work under the direction of Bruce Lamb and I. And I'd like to thank our collaborators as well. And thank you for paying attention. And